Hello everyone, welcome again to Learn Science with Neha. We'll be continuing from endoplasmic reticulum. Let's start taking colorful notes and making our science more interesting and more interactive. So this was our last topic of endoplasmic reticulum, which is like a framework through which all the transportation of various things like proteins and lipids is happening. Along with it, various biochemical reactions are also happening because it is creating a base of it. Now we'll see it forms. Like how many forms are there, like how is it arranged. Now, if we talk about endoplasmic reticulum, you will see it has got three different type of structures in itself. So, number one, the first structure you call it cisternae, okay, which is like a sac-like structure piled from over one another. So, this is first cisternae, second cisternae, third cisternae, like this. And the second one is the vesicle. Vesicle is circular structure, and the tubule, which is like a tube-like structure. So there are three different kind of structures of endoplasmic reticulum. Now we are moving towards Golgi apparatus. Golgi bodies, which we'll be doing in the next slide, and Golgi bodies again have got two type of structures. Number one is cisternae, as I told you, sacs piled over one another like this, and these vesicles. These are the secretory vesicles which are in circular form. So next is vacuoles. So vacuoles, if you know, vacuoles are of they are present in plants also, in animals also. But in animals, vacuole size is very small because we don't need much of storage in our body. But in plants, the vacuole size is very big, rather bigger than any other organelle in the plant cell. So vacuoles provide turgidity to the plant cell because they have to stay outside in a hot weather. Now let's read about vacuole. So vacuoles are basically storage sacs. They store liquid and solid content. They are smaller in animal and bigger in plant cell. Now very interesting. So if you see vacuole is bilayered, double membrane, and this membrane is called as tonoplast. Okay. So if we talk about what is there in the vacuole, you will see salts, minerals, pigments, and proteins. They are there. So this all combined together to form a cell sap. So that you might have heard in plant cell ascent of sap. Ascent means movement in the upward direction of sap. Sap means water, minerals, proteins from the roots to higher parts. That is ascent of sap. And that sap is collected in the vacuoles. Now, it provides turgidity and rigidity to the plant cell because vacuoles provide much of the space, and here in the vacuole, if you see, so whatever water is there, water is exerting pressure on the outer wall. So this will prevent the plant cell from collapsing. So for example, you have got water balloon. So if you hit your body with a water balloon, your water balloon during the Holi, so it will actually pain. But if you hit anyone with an air balloon, it will not pain because water itself is creating a turgid pressure in the balloon. So this is called as vacuole. And if you talk about amoeba, in amoeba, vacuole has got a slight different function, that it helps in storing food and also expelling the waste material outside the cell. Now, after vacuole, we have got Golgi bodies, which is of two structures are of two types, cisternae I told you, and the vesicle. So Golgi was discovered by Camillo Golgi, from where it got its name. It is again membrane-bound vesicle containing sacs one over the other, which is called as cisternae. So Golgi bodies have a very beautiful function like gift packing. You go to any shop, you say, I need to pack my gift. So it will take the gift, pack it beautifully and give it to you. Same is the case with Golgi bodies. So whatever proteins are formed by endoplasmic reticulum, they are passed to Golgi bodies. They are packed properly and then they will move like this, and then they are transported maybe outside the cell, or if it is required inside, it will go inside. So basically the function of Golgi bodies is to pack and transport the protein. Plus along with it, Golgi bodies have major function of creating lysosomes. So lysosomes are suicidal bags. Along with it, it makes complex sugars out of simple one. You might have heard in our body complex sugars are broken down into simple one, like starch is broken down into glucose. But here it is opposite. It forms complex sugar. So extra sugar which is there in our body, it is stored in the form of glycogen, and that is made by Golgi bodies. So Golgi bodies form connection with endoplasmic reticulum. So that's how the transportation can occur. So now lysosomes, which are also called as suicidal bags, as they contain hydrolytic enzyme in them, which will break any unwanted substance or any worn out organelle and will protect our body from the poison which that organelle could have caused in our body. Okay, so the organelle is called, the enzyme is called as hydrolytic enzyme, and Golgi bodies are also called as waste disposal system. So they are called as suicidal bags because sometimes, some organelles or some cells become very, very toxic, very poisonous. So they will kill themselves along with the cells so that our body is protected. That's why they are called as suicidal bags. Now, very, very important, mitochondria, which is a semi-autonomous body of the cell. Semi-autonomous means it has got a capacity to produce its own DNA, which is very, very important, and own ribosomes. So these are the two major things which are required in the cell. So it can produce its own, that is why it is called as semi-autonomous body. Along with it, mitochondria is powerhouse of the cell. It is double membraned organelle. Okay, it produces ATP, and it has got again two type of structures. Number one, you have got your inner structure. Outer is the capsule, if you see, and inner again the cisternae. Cisternae are there to increase the surface area on which ATP can be produced. So they increase the surface area for production of ATP. So this is mitochondria, also called as powerhouse of the cell. Now let's check our knowledge again. So the question one is, you will get this kind of cell. So outside it's a capsule, you can see, and inside are the cisternae. So this organelle states that it is mitochondria. Okay, so what is X and what is Y? So X are the inner parts, which can be cisternae, and Y can be a capsule. Okay, so let's see. So X is cisternae and Y is the matrix, that is the base in which cisternae is there, in which all the energies are being produced and biochemical reactions are happening. So why is X so many folded? So that it can increase surface area of absorption, and it is mitochondria. Now if we talk about plastids, they are present only and only in plant cell, and this diagram of plastid is very important because it generally comes in exam. It has got granum, so placed one over the other. Okay, so plastids are present in plant cell. It is of three types: chloroplast, chromoplast, leucoplast. Chloro means green color, so it helps in the production of food with the help of chlorophyll, which is there inside it. Photosynthesis happens. Chromoplast gives color to the flower so that insects can be attracted and pollination can occur. And leucoplast, it stores starch and protein which is being produced by the plant cell. Now comes these questions which you have to answer me in my comment section below. Let me see who can answer. WBC is also a single cell as amoeba, but how are they different? Second question, which part of the cell provides it shape? This I can give you a hint. Shape can be rigidity, so it can be either cell wall or vacuole. You have to choose one. Number 3, cell wall is additional covering that provides shape and rigidity to the cell. Which other organism besides plant cell have cell wall? So the same thing I have given you in my part 1, you can see. Except plant cell, which two other organisms have cell wall present? And the last one, besides cell wall, which other component in plant cell supports rigidity? We just did now. Which is the organelle which provides rigidity due to its turgid pressure? So give me the answer. Now comes the next part. Difference between plant cell and animal cell. Plant cell has got cell wall, number 1. Animal cell does not have cell wall. Plant cell has a large vacuole. Animal cell has very small or no vacuole. Plant cells have plastids. Animal cells do not have plastids. So these three things are there, and rest you should know what is a diagram and proper labeling. Now again check your knowledge. So here you have got the organelle. You have to label W, X, Y and Z, which is a covering of membrane X. So if you see X here, so X is the largest membrane. This is how you have to rule out few of the options. So X is the largest body. So largest body, it is obviously a plant cell. This makes, it is a plant cell. So largest body can be of vacuole, and the covering of vacuole I told you is tonoplast. Okay, this one is vacuole, understood. Now comes, which of the organelle contain hydrolytic enzyme? Hydrolytic enzyme I told you, they were lysosomes. So lysosomes contain hydrolytic enzyme, and which of them take part in storage, modification, packing? That I told you, gift packing, that is Golgi bodies. So now you have to tell me which is W, X, Y and Z. I can give you answer here. Okay, and the answer is W is Golgi bodies, X is vacuole, Y is ribosome, Z is lysosome, and the covering of membrane is tonoplast. This completes your chapter, and these are the two questions which you can give me in my comment section. Number 1, you have to give me labeling of A, B, C and D, and number 2, you have to tell me 2 organelles each. Number 1, which is having no membrane, single membrane or double membrane. So this finishes your chapter. Hope you have understood well. Do like, share and subscribe to my YouTube channel and press the bell icon to get the regular